How's it going, everyone? Welcome back to my channel, and today we finally have the update to our PCD apoptosis program cell death. In our last video we discussed the intrinsic pathway of apoptosis, which deals with the signal or death of a cell coming from the inside, some sort of death stimulus from the inside. Now we're going to go into a little bit more of a more difficult pathway to kind of analyze, because this actually can diverge into the intrinsic pathway, as well as this standard mechanism. So to deal with it again, to recap, apoptosis, PCD, program cell death, and in this pathway, the extrinsic pathway, we are dealing with some signal that's coming from outside the dying cell. Usually the signal comes from another cell. So in here, I drew right here is a little bit smaller than the dying cell right here, because it has a lot of stuff that we're going to discuss as well with this formation. But let's say here we have some sort of lymphocyte. Lymphocyte is part of the immune system, which consists of mainly these three. You got a killer T cells, natural killer T cells, and B cells. These are usually used to combat viral, bacterial, and other pathogens. Natural killer T cells can also fight against cancer cells as well. But let's say, for example, we have a cytotoxic killer T cell as our example. In here, it has this little thing that's kind of sticking out. It's meant to be some sort of ligand. So a ligand can either come into some sort of metal ion or some sort of element, but it can also be any type of substrate that's binding to some sort of receptor or an enzyme. In this case, we have our death receptor of our dying cell that's going to bind, that the ligand's going to bind into. Some of the commonly known receptors that we know in apoptosis involve FAS, tumor necrosis factor, and TRAL. These come in all kind of pretty much belong in the family of the TNF, and they all have their own specific death ligand that binds into them to start this pathway. Now that we got that, let's see what happens. So the ligand will, from pretty much the killer T cell, will just straight up tell the cell, okay, you're about to die, or bind to this death receptor. Let's say the death receptor is FAS. That's the most common one that we use. The other ones have a little bit more intricate. Once it binds in there, it has this, you can see this receptor, it kind of draw it weirdly. It's supposed to be a trimerization of this receptor, which involves the activation and recruitment of FADD, or FAS associating death domain, and caspases. This one has its own caspase that gets recruited that is different to the intrinsic pathway. So this entire pathway deals with just recruiting all of these type of death domains, and then the main point is this formation of DISC. This stands for death inducing or induced signaling complex, which once it pretty much is completed once you have your inactive procaspase ACE finally recruited with the death effector domain coming from the FADD adapter protein. This is all pretty much involved in the whole signaling transduction pathway of the extrinsic apoptotic pathway. Okay, now before we actually discuss kind of like this is just some random stuff that I thought was interesting, but for the most part, the ending of the extrinsic apoptotic pathway, we have the recruitment of procaspase 8 in part of the death inducing signaling complex. Becomes activated as caspase 8 through some sort of proteolytic cleavage of this procaspase. Then it recruits again with the whole caspase that happens. It recruits procaspase 3, activates it as caspase 3, which then fully initiates cell death via the extrinsic pathway. On the top here, there was some things that were involved. If we go back to the depth domains that we discussed in the formation of disk, there were several things that I thought was pretty interesting. So we got the depth domain, which was used, that was part of the depth receptors, as in like FAS or FOS, that is used to recruit FADD. These depth domains then, as they recruit FADD, FADD has the depth effector domain, which recruits the pro-caspase. Many of the caspases have many of these motifs, specifically the depth effector domain, which is just used to interact with other proteins in the assembly of disk. Another part of it that's also used is called caspase activation recruitment domain and the prion domain. These are tertiary structures. So in proteins or other enzymes, you have primary structures, secondary structures, and coordinate structure, tertiary structures and coordinate structures. So with the basic primary structure of amino acids, you have pretty much a listing of your 20 amino acids, kind of in this really small, basic polypeptide chain. Secondary structures involves the folding of the primary structures that we got, producing these things known as alpha helices or beta sheets. All of them have their own type of configuration, and mainly is usually stabilized due to the amino groups and the carboxyl group COO minus, which has actually these interactions of the folding. Whenever you have these multiples of these types of secondary structures, you have your tertiary structures, usually having this single subunit, and then the coordinate structures when you have multiple subunits. These depth folds are kind of structural motif, where in biochem we kind of discuss these types of weird things like zinc fingers, helix loop helix. There's these kinds of structures that make up the more higher level protein structures. And this one is an example of a Greek key motif, which kind of has this, I don't know if you can see the arrows. This is meant to show as it kind of goes a repeating anti-parallel. So it goes from one direction and then goes the opposite, go back the other direction, opposite. This one's a little bit mixed. So we have the C terminus right here, and then the N terminus. And then with the depth folds, they mainly consist of alpha helices anti-parallel to each other. So I kind of do it in the way like we got the one going in the, say like it's right-handed, the other one going left-handed, kind of repeatedly until the end. And so this is kind of like around 6 alpha helices. 1, 2, 3, 4, 5, 6. So yeah, this is mainly what these depth folds look like, and these four domains consist of these. So like I said, a lot of these proteins, I mean, they're a lot more bigger, but these are just particular subunits that they have. Kind of the last bit that we got here regarding the intrinsic pathway. There's another type of, I guess, antipatotic protein, and it comes as the word flip, and I'm sure I should have capitalized it, but they are proteins related that can inhibit the formation of disk, which is the very beginning of the intrinsic pathway. So you can have the whole ligand binding to the depth receptor, but the depth domains do not recruit any of the sequential proteins needed for the caspase to occur. Another thing, that whenever procaspase is recruited with the depth effector domain coming from FADD, it also, whenever there's the cleavage part to activate it, there's also a cleavage of this bit protein, which is actually related to the pro and antipatotic proteins involved in the intrinsic pathway. It becomes truncated. It's known TBID, or truncated BH3 interacting domain agonist. This plays a huge role, and not a lot of people actually talk about it when talking about apoptosis, but TBID has a weird function where it promotes the mitochondrial outer membrane permeabilization that is involved in the intrinsic pathway. It has the ability to inhibit any anti-apoptotic proteins such as BCL and promote BAX and BAC. These are the pro-apoptotic proteins that make the pause inside the mitochondria, releasing cytochrome C, and then y'all know what happens there. Cytochrome C is the main thing that makes this apoptosome, essentially, thus initiating the death of the cell with the intrinsic apoptotic pathway. That's why I kind of want to discuss the intrinsic first, because this actually also plays a huge role in also activating it in this weird manner as well. So if a cell doesn't die with this pathway, it has this as another way to do so as well. But yeah, this is pretty much the end of the basics of apoptosis and the two pathways. In the following video, we'll be discussing how to detect apoptosis in cells with particular assays that are used in scientific journals. Hope y'all enjoy this video, and I will have the video on the intrinsic pathway in the description if you want. It's a good video to kind of explain the importance of apoptosis, as well kind of give you an idea of how these cells kill themselves and why it's really important, especially in cancer biology. And yeah, hope y'all stay safe and have a great day. Take care.